Hello.
Mo ní kò lè possible mo ń ṣe nǹkan lórí fóònù mi mo ń bí o.
Was the time that I trained. It's not fast.
Hello, good morning.
This is very fast. Àdúrà fún màjú mi sàn. Lọ́ọ̀tù.
Good morning.
Are you ready?
Okay. Can you all hear me?
I did not hear. I finished.
Morning. Can you hear me?
Good morning, Master.
So we will
Yes,
start.
ma'am, we can hear you. Good morning.
All right. So last week started by talking about the cell, and we looked at what it is.
Only when I'm
That, good morning. Can you all hear me, please?
yes, ma'am.
Yes,
Yes,
ma'am.
ma. Go on, please.
Okay. So last week it is
Yes,
not
ma'am.
my. My name is Olumide Adebesin. Last week we had a class, and I've sent the slides. I hope you have seen it, where we talked about the cell, and we looked at the cell, and we tried to define what a living thing is, and we agreed that the cell was the structural and functional unit of life, and so anything that has life must be made up of cells. Therein, we said each cell will have genetic material, a cytoplasm, and organelle. Now, there are two different types of cells. The cells that have their genetic material not enclosed in a membrane, those type of cells are called prokaryotic cells. Pro means before, karyo means nucleus. And those cells that have their genetic material surrounded by a membrane are called karyotic cells. Eu means true, karyo means nucleus. So we looked at the historical perspective, the we looked at the scientists. We looked at the scientists that worked hard to give us what is called the cell. And then we also looked at the organelles. The organelles that are that don't have membrane. Organelles are special bodies within the cell that perform specific functions. And we can classify the organelles into three main groups. The ones that do not have a membrane surrounding them, which is an example is the ribosome, and the ones that have membrane about them, and then the ones that have double membrane. Those that have double membrane around them include the nucleus, the mitochondria, and the chloroplast. Yes, and I made an analogy when I was trying to describe the cell. I made an analogy when I was trying to describe the cell, that the cell, we can liken it to a pot of soup, where the pot and the cover is the cell membrane, and everything inside the pot, the soup itself, is the cytoplasm. And I did say, okay, somebody made the egusi soup for us that day. And I did say, if you took the pot of soup and you turned it into a strainer, then what is retained in the strainer are the solid parts, which is the organelle. And what is strained out is the liquid part we call the cytosol. So we concluded that the cytoplasm is made up of cytosol, which is the fluid part, and organelles, which is the solid part. And then I went further to say, if you add one hard-boiled egg into your soup, that hard-boiled egg will be the nucleus. Please, Emmanuel Divine, mute yourself. Emmanuel Divine, mute yourself. Don't be careless, please. Mute yourself as you yeah, thank you. So I said if you added the hard-boiled egg to it, then that hard-boiled egg represents your nucleus. Now, herein lies the difference. some cells have cell wall. So if you put a pot in a pot warmer, that pot warmer will be your cell wall. So prokaryotic cells do not have the egg, okay? And they have the cell warmer, okay? The pot warmer rather. So that would mean they have cell wall. And cell wall in prokaryotic cells are made up of peptidoglycan. In eukaryotic cell, you can divide them into two, the plant cell and the animal cell. Eukaryotic cell, the plant cell has cell wall, okay? Has cell wall. Cellulose, the one that is made of chitin. Animal cells, cell wall. All right. We'll look at those maybe in. All right. You see my screen. It is a structure showing the animal cell. And the animal cell will not have any cell wall. It has a plasma membrane. Plasma membrane, cell membrane is the same. All right. And it has it's showing all the organelles, majority of the organelles that are within the animal cell. All right. Another view of the animal cell. So the outermost part of the cell, which is the pot in your soup, is called the plasma membrane or the cell membrane. Okay. Now, this plasma membrane is selectively permeable. That means it selects the materials it allows to go in or out of the cell. And the plasma membrane is made up of lipid bilayer. And that lipid bilayer has proteins inside it and on top of it. Okay. The plasma membrane is present both in plant and animal cells. It's also present in prokaryotes. All right. Now, I know you have done biomolecules where you were exposed to phospholipids. Okay. So this plasma membrane is composed majorly of phospholipids. All right, with proteins inside it, and some proteins are attached on top of it. So we can have integral proteins and peripheral proteins. Those integral proteins span at least one leaflet or both leaflets of the plasma membrane, while the peripheral proteins are attached to either a protein that is embedded in the plasma membrane or loosely attached to the surface of the phospholipid bilayer. Also, you have another group of proteins which we call lipid anchored proteins. Those ones look like balloons, balloon with a rope that used to hold the balloon. So it's a lipid that holds the protein to the cell membrane. So based on the structure of the membrane, it is regarded as the fluid mosaic model because many scientists in the beginning proposed a lot of theories or hypotheses on how or what they believe the cell membrane should look like. And the one that was acceptable or that is still acceptable is the fluid mosaic model. Okay. So the functions of the membrane. The membrane is selectively permeable. That means it permits the entry of selective materials in and out of the cell based on the cell's requirement. Now, in an animal cell, the membrane functions to shape and protect the inner contents of the cell. So, but in plant has an additional cell wall which protects, and bacteria also has additional cell wall which protects also. All right. So here is a image of the cell membrane where you have, of course, the okay, where you have the proteins, different types of proteins, okay, and also have the carbohydrate chains attached to proteins. Now, your red blood cell, the red blood cell we have in our body, our blood group is determined by these carbohydrates. that are attached to the surface of the red blood cell. So whether you are blood group A, B, AB, or O, depends on which type of carbohydrate is present on the surface of your red blood cell. And underneath the cell membrane is the cytoskeleton. All right. Okay, so here are a few of the proteins you will find on the cell membrane. You have enzymes, you have storage proteins, you have transport proteins. Okay, and of course, you have antibodies. You have contractile proteins also, right? And structural proteins amongst the few. And of course, hormones. Hormones, because they serve as signaling molecules and they help us achieve cells. Now, some of those proteins act as receptors. Receptors in the sense that they are there to receive molecules, what we call signaling molecules, that make things happen in the cell. Now, there's a transport process, all right? And the transport process just simply is telling us how things cross the membrane. And things can cross the membrane using in a passive process. That passive means that no energy is required. It can be by simple diffusion, facilitated diffusion, osmosis, or filtration. The active process actually involves some means of transport. Okay, so that means things are moved across the cell by proteins, and that means energy is involved, expenditure of energy. So we have different types of transport systems. You have the uniport, the symport, and the antiport. Uniport means that the protein is moving a molecule in one direction, whether it's moving it into the cell or is moving it out of the cell. Symport means that the molecule is moving two things in the same direction, whether simultaneously or sequentially. What that means is that please kindly mute yourself once you come in. Mute yourself, please. Temilade Wealth, mute yourself, please. Then Neka Onyechi, your question. Temilade Wealth, please kindly mute yourself. Neka, your hand is raised up.
Yes, sir. Yes, sir. Yes, sir. Good morning, sir. Good morning. Because
Yes.
during the last class, we stopped at the last class we stopped at microscope. Okay, so I wanted to ask, this class we're having now, that's a plasma membrane and all this transport processing. Is it after the microscope? Because I'd already made notes based on the last.
I'm sure you have the slide. Please mute yourself. I'm sure you have the slide I sent. Did you get the slide that was sent for that class?
No, sir. I didn't get it, sir. I'm just but I wanted to know if this class is after the microscope because I already made the notes.
Madam, the class is a continuation from the last class and it's yes after the microscope because
Okay, sir. Thank you so much, sir.
All right. So you can drop your hand so that I don't think there's somebody there again. Please. Thank you. All right. Okay, where were we? Okay, so the uniport transport system is just saying that a protein, which we regard to as a transporter, located in the membrane, is going to move one product, one substance, either from the outside of the cell to the inside, or from the inside of the cell to the outside. The symport type of transport, and the protein that does symport is called a symporter. That protein will move two molecules into the cell at the same time, or one after the other, or it will move two molecules out of the cell at the same time or one after the other, immediately after the other. Now, the antiport transport system, that protein that does that is called the antiporter. That protein will move two molecules in opposite direction, either at the same time or sequentially. That means if it's moving one molecule out of the cell, it's bringing in another molecule, either at the same time or just one after the other. So you have the primary active transport and you have the secondary active transport. So it's an active process and that involves bulk transport, which is vesicular transport. So you have endocytosis. Endocytosis means bringing things into the cell. If it is, example is the pinocytosis, which is bringing liquid into the cell. Okay, I'll increase my voice level. So endocytosis is bringing things, endocytosis means bringing things into the cell from the outside of the cell. Example is pinocytosis specifically points to bringing in liquid into the cell. Phagocytosis is bringing solid materials into the cell. And receptor-mediated endocytosis means there's a protein on the surface of the plasma membrane that binds a molecule and brings it into the cell. So the cytoplasm, that cytoplasm is everything inside our pot, okay, minus the cell membrane and the nucleus. Everything inside the pot minus the pot itself and the hard-boiled egg. Hard-boiled egg represents the nucleus. The pot itself represents the cell membrane. All right. So the cytoplasm is present in bacteria, plant, and animal cells. It's a jelly-like substance. Yeah. And it's composed mainly of water, organic and inorganic compounds. It is the site for chemical reactions within the cell. That's where majority of the metabolic activity takes place. Now, another organelle that's present in both prokaryotic cell and eukaryotic cell is the ribosome. The ribosome is a non-membrane bound. That means the organelle is not surrounded by a membrane, but it's a very important organelle. It can be free floating within the cell with cytoplasm or associated with endoplasmic reticulum. Okay, it is composed majorly of ribosomes, ribosomal RNA, that's rRNA, and ribosomal protein. Okay, so prokaryotic ribosome is 70S. S there is feedback unit, is a unit of intensity. Okay, now the eukaryotic ribosome is 80S. Those ribosomes are the intact ribosome for prokaryotic is 70S. The intact ribosome for eukaryotic is 80S. Okay, and the ribosomes are made up of two subunits. Okay, the small subunit and the large subunit. The small subunit and the large subunit. For the prokaryotic ribosome, the small subunit is 30S, and the large subunit is 50S. So, but for the intact ribosome, where you have the small subunit and large subunit together in the prokaryotic, it is 70S. For the eukaryotic ribosome, the small subunit is 40S and the large subunit is 60S. But the intact ribosome, which comprises of the small and large subunits together, is 80S. All right. So they are involved in amino acid polymerization. That means they join, they synthesize, they are the site where polypeptides are synthesized. Now, the endomembrane system. The endomembrane system regulates protein traffic and performs metabolic functions in the cell. And that the endomembrane system includes different structures. Okay, so we have the endoplasmic reticulum and its vesicles. We have the Golgi apparatus and the vesicles. We have the lysosome, and then we have the vacuoles. Endoplasmic reticulum, that's an organelle. There are two types of endoplasmic reticulum. You have the smooth endoplasmic reticulum and this rough endoplasmic reticulum. Then there's the Golgi apparatus or Golgi body. Then there are the lysosomes, the vesicles, and then there are the vacuole. I hope we are still following. Good. All right.
Yes, sir. Yes, sir.
Yes, sir.
Thank you.
Yes,
So
Yes,
here
sir.
is
Yes, sir.
Yes, sir.
all right. Thank you. Thank you. Thank you. You can mute yourselves. Okay, good. All right. Now, the endoplasmic reticulum. What's the function? It manufactures membrane and performs very many biosynthesis functions. Okay. So it is a membrane connected to the nuclear envelope. Now, the nucleus has two membranes, two lipid bilayers, two of them surrounding it. And the two are pinched off at a space. That space, we refer to it as nuclear pore complex. That's where things go in and out of the nucleus. So the space between the two membranes that make up the nuclear envelope is continuous with the lumen, that is the center of the rough endoplasmic reticulum, right? So the rough endoplasmic reticulum looks rough under the electron microscope because ribosomes are attached to it. The smooth endoplasmic reticulum does not have ribosomes bound to it, so it looks smooth under the electron microscope. So what are the functions of the smooth endoplasmic reticulum? They function in metabolic processes, which specifically is synthesis and hydrolysis. And they are tube-like structures, okay? Enzymes of the smooth endoplasmic reticulum will synthesize lipids, oils, phospholipids, steroids, and sex hormones. That's steroidal hormones, yeah. And then they partake in the hydrolysis of glycogen to glucose in the liver. The liver is an organ in the human body or in body of animals, okay? And the liver, when you take anything into your body, it's the liver that gets it first. So if you have taken any poison, it will go to the liver first. If you have taken food, after it is digested, you go to the liver first, okay? So the liver detoxifies drugs and poisons, okay? And then, of course, it pumps calcium ions for muscle function. That is the rough endoplasmic, the smooth endoplasmic reticulum. In muscle cells, it serves as a storage for Ca2 plus ions. Rough endoplasmic reticulum, it produces protein for export out of the cell. Now, this is very critical, and it can achieve this function because it has ribosomes attached to it. And ribosomes are the machinery that produces polypeptides, which fold up to become proteins. And whatever it produces is packaged into vesicles, what we call transport vesicles. We have transport vesicles, we have secretory vesicles. So what happens is when these ribosomes mix the polypeptide into the lumen, into the center of the rough endoplasmic reticulum, those things are engulfed in the membrane of the rough endoplasmic reticulum and pinched off. So rough endoplasmic reticulum synthesizes membrane phospholipids. So it builds also new membranes because remember, whatever protein is made into it, it is budded, it is pinched off and surrounded by a membrane before it is transported to other parts of the body. Now there's another organelle called the Golgi apparatus. The Golgi apparatus serves it finishes, sorts, and ships cell products. So products coming from the rough endoplasmic reticulum products coming from the smooth endoplasmic reticulum in vesicles fuse with the Golgi apparatus. And then the Golgi apparatus checks these products. If these products do not meet the standard, the Golgi apparatus sends it back to the to whichever, whether the smooth endoplasmic reticulum or the rough endoplasmic reticulum. So what happens is the Golgi apparatus, apart from being a central post office, is a good ping pong player, table tennis player. So when smooth endoplasmic reticulum sends materials to the Golgi in form of transport vesicles, and they fuse with the cis face of the Golgi apparatus, Golgi will look at those things and process it. If they are not okay, it is going to board it off again as a transport vesicle and send it back to smooth. Or if it needs rough endoplasmic reticulum to add anything to that thing, it will send it in form of a transport vesicle to rough endoplasmic reticulum. And this process goes on between smooth endoplasmic reticulum, rough endoplasmic reticulum, and Golgi apparatus until Golgi is satisfied with the product. Then it now finishes the product, it sorts it, and it labels the product. When it labels, it allows to send the product to where it is needed. That is the shipping and receiving shipping. It sends it to where it is needed. When it receives, it receives. So Golgi is very important in the cell. So it is a flattened membranous sac-like structure. So it looks like pizza bread. You know, this bread used for making shawarma. There's this type of bread that is flattened sack, and you have to open the inside and throw your things in to make shawarma. So it looks like that. It's stacked like that, but it has a face. The face that is adjacent to the endoplasmic reticulum is called the cis face of the Golgi. Cis means same side, and then on the other side is the trans face. So vesicles, transport vesicles come in from the cis face of the Golgi and leave from the trans face of the Golgi. So during the path from cis to trans, products from the ER are modified into their final form. Tags, salts, and packaged materials are into transport vesicles. So the Golgi can be regarded as the general post office headquarters. And then the transport vesicles are delivery trucks. So delivering package will have stamps or barcodes on it, and so the delivery trucks know where to send those packages. Lysosome is another membrane-bound organelle that is boarded off from the Golgi. It is boarded off from the Golgi. And lysosome contains hydrolytic enzymes. And these hydrolytic enzymes digest most of the macromolecules. So the lysosome is known as the waste disposal organelle of the cell. Okay, it functions as a little stomach. That means it can digest most things. So lysome means breaking things apart, and some means body. So it is the cleanup crew of the cell. So lysosome, please note, lysosomes are Golgi-derived vesicles, and they contain digestive enzymes. You find lysosome only in animal cells. So how does lysosome perform its work? Lysosome can fuse with food barcodes, and digest the contents, and the contents are released into the cell. Okay? Autophagy. That means lysosome, when it detects, because if organelles within the cell are not working properly, they are tagged for destruction. The lysosome will fuse with it and digest those organelles and then pass the digested product to the cell. Again, lysosome can do when the cells bring these things by phagocytosis. Like lysosome will fuse with the phagosome and digest whatever it is. Now, lysosomal enzymes work best as a pH of 5.0. The internal pH of the cell is 7.2. So this is very important that the pH of lysosomal enzyme is 5. Reason is because, take for example, if lysosome, the cell membrane is broken by whatever means, then it means all these digestive enzymes hydrolytic enzyme will release it to the cytoplasm, and they will end up digesting everything in the cytoplasm. But the pH in the cytoplasm is not favorable. Is 7.2. But these enzymes from lysosomes can work best in acidic pH. So you don't want to digest the cell doesn't want to digest itself. So in case there's any leak of enzymes from this lysosome to the cytosol, it will not function properly. But when things go wrong, example, when the digestive enzyme in lysosome does not work properly, then it means the lysosome will be filled with undigested material and just go larger and larger and disrupt the cellular function, the function of the cell. And if this function of the cell is disrupted, then it means that eventually the function of the organ from which tissue the cells are made, it will be disrupted. So basically the cell will be perturbed. So there is a Tay-Sachs disease, is a lysosomal storage disease, which is usually fatal. And it's the condition is identified by lipids building up in the child's brain, and the child probably die before the age of 5. Now, peroxisomes. Peroxisomes are another membrane-bound organelle that house digestive enzymes. Remember, lysosomes house hydrolytic enzymes, but peroxisomes will house, oxidative enzymes. All right, and they are present in both plants and animals. Okay, oh, sorry, this is supposed to be oxidative enzyme. Yeah, pardon me. All right, so what they do is they are involved in power generation, and they're also involved in killing things that the cell does not want, because they have oxidative enzymes, and they end up producing hydrogen peroxide, which is key, because this one can break down bacteria. Okay, so they are also charged with detoxifying alcohol and other poison. Okay, so we have this vacuole. In plants, you have a large central vacuole, but in animal cells, you have small vacuoles. All right, vacuoles are large water-filled spaces. Okay, in plants, it's cell sap that is there, but in animals, you can have vacuoles that can store small things. And the vacuole is surrounded by a single membrane called tonoplast. The membrane that surrounds the vacuole is called a tonoplast. Okay, so the vacuole is used for stockpiling proteins or inorganic ions. And it can also be used to store red and blue anthocyanins, acid, salts, and so basically vacuoles, when you see brightly colored fruits or things like that, those colors are stored in the vacuoles. All right. It's also a sac for depositing metabolic byproducts so that it doesn't harm the cell. Okay, so it can store pigments, and it stores defensive compounds against herbivores. So that's why you see some herbivores. Herbivores are animals that feed on plants only. You see some herbivores will not go near certain plants because they know that it houses some poisons that is harmful to them. All right. And the vacuole also in plants help maintain tug-up pressure. So when the vacuole doesn't have enough water, then the plant wilts. Okay. So you can have food vacuoles, which are formed by phagocytosis, and you can have contractile vacuoles. These are charged to pumping excess water out of protein. So the endomembrane system is a complex and dynamic player in the cell's compartmental organization. So let's look at the flow pathway, the pathway of membrane flow. So basically, the nucleus, remember I told you, has a double plasma membrane. Okay, and the inside of the space between the double plasma membrane, the space in the nuclear envelope, That double membrane is called nuclear envelope. The space there is continuous with the lumen that's center of the endoplasmic reticulum. That's rough endoplasmic reticulum. And remember, rough endoplasmic reticulum is connected also to the smooth endoplasmic reticulum. The smooth just differs from rough in that rough has ribosomes on the surface, a lot of it. And then all these interact with the Golgi apparatus, okay? And the Golgi apparatus also makes lysosomes. And then don't forget, you also have vesicles, transport vesicles or secretory vesicles. Those transport vesicles are those vesicles that move things from one organelle to the other within the cell. But the secretory vesicles are those vesicles that will move things from within the cell to the outside. And of course, then you have the plasma membrane, and this makes up the endomembrane system. So the nucleus. Now, we have been talking about membranes that have a single plasma membrane around them. Now, we'll talk about organelles that have double plasma membrane around them. All right. The first one we'll be talking about will be the nucleus. The nucleus is a double membrane organelle and it's found in all eukaryotic cells. Remember, prokaryotic cells do not have a membrane-bound nucleus. Instead, they have a region within the cytoplasm that we call a nucleoid. It is not that the genetic material is not surrounded by a membrane. Okay, so
so you don't come down to 150 for you like this.
Please mute yourself. Oh, sorry. Soluwani Ogulade, you have a question? I'm sorry, I wasn't paying attention.
Yes, sir. I wanted you to come again in the part that you said peroxisomes house oxidative enzymes. You also said the lysosome house. What do they house, please?
Lysosomes house hydrolytic enzyme.
Okay, thank you, sir.
Hydrolytic. That part of the peroxisomes is supposed to be oxidative, not digestive. So lysosome is one that houses digestive. Because hydrolytic enzymes, what they do is they break bonds by adding water. And you have studied biomolecules, if I'm correct, and you have been shown how elements come together to form the molecules and how the molecules come together to form polymers, how monomers come together to form polymers.
Yes, sir.
Yes? Okay. Owen.
Good morning, sir.
Morning.
I have a question. You said that when materials are sent to the Golgi bodies and the Golgi bodies reject those materials, maybe the materials are wrong, which part would it take back to the endoplasmic reticulum? Is it the cis side or the trans side?
No, it's everything, whatever it's coming is received by the cis. It's the trans that sends things. But if the material is not okay yet, the Golgi checks it and then it busts it out from the side and sends it back.
Okay, sir.
So it's like playing table tennis with themselves until Golgi is satisfied. The one Golgi can modify it to modify. When he modifies it and he needs the plastic rough to add something to it, it will push it back via transport vesicles. That's why they are called transport vesicles. But if Golgi has is satisfied what is there, like in the process or in the case of lysosome, the lysosome will burn it out from the transphase. If it has to send things outside the cell, it will put it inside secretory vesicles. Okay, so transport vesicles do perform another function, different function from secretory vesicles. Transport is to move things within the cell. Secretory is to take things from the cell or from any organelle and push it outside the cell via the process of exocytosis. All right. Yes. Festus, your hand is up. Okay, then chick chick Chicamzoderic.
Sir, I like more explanations on what happens to the lysosome when it becomes damaged.
Okay,
Yeah.
lysosome, when it becomes damaged, it is going to
Yes, sir.
leak. It's going to leak its contents, which are hydrolytic enzymes, into the cytoplasm. But those hydrolytic enzymes, they function. The optimal pH is 5.0. Okay,
Okay.
that's where they
Okay.
work best. But in the cytoplasm, the pH of the cytoplasm is 7.2. And it's designed that way because if there's leakage of hydrolytic enzyme into the cytoplasm, then those enzymes won't function properly. If the converse were the case, that it was very active at pH 7.2, then once there is that leakage, it will digest the whole cell, the content of the cell.
Okay, sir.
Lawal, Lawal. Okay, sorry. Okay, just you're not you have muted yourself, so it's okay as long as you're not making noise. All right. So I'll go on now. Derek, can you please drop your hand? Thank you. Derek, please drop your hand, please. Princess.
So, drop what's up?
Yes. Good
No.
morning, sir.
Morning.
Okay.
Yes.
Yes, sir. You said something about, is it the lysosome that houses the hydrolytic enzyme?
That's correct.
Okay, because you also
And the
said
peroxisome
okay.
house oxidative enzyme. I told you that was an error on my part when I wrote digestive enzyme.
Okay, sir.
Okay. Peroxisome is charged with power, generating power, because it's oxidative. Okay. There's something we call them what have you heard of oxidative stress, free radical generation?
No, sir.
Okay, no problem. Just know that peroxisomes have, or within them, they house oxidative enzymes.
All right, sir. Thank you.
And peroxisomes are not boarded up from the Golgi. Peroxisomes do not originate from the Golgi. It's only lysosome that originates from the Golgi. Okay?
Yes, sir.
Peroxisomes originate directly from rough endoplasmic reticulum. Okay. All right.
All right. Thank
You're welcome. Okay, so nucleus. I said it's a double. Okay, so how many of you have gone to, say, a bakery to buy bread, hot bread, and then you have been given your bread, and they now give you a nylon, and you struggle to open that nylon. Then you have to maybe ring the nylon and then blow, and then the nylon separates into two. Okay, so that nylon as it is before you open it is your nuclear envelope. Now you know there's a space. The space, if you add there's a space within the two parts of the two sides of the nylon are two plasma membranes that are pinched off at the ends to form your nuclear envelope. Okay?
So I can't see your screen again.
Okay, just okay. These guys, you people have done it. I hope you have not logged me out.
No.
No, sir.
No, sir. Sir, please.
You're still in the meeting, sir. Just share your screen again, sir.
All right. Let me just go here. Okay, just connecting back. Oh, network. Okay, so you can ask questions in the meantime before I get it back up.
Sir, please, I have a question.
Yes, please ask your question.
So, sorry, please, in the previous slide, I was confused.
Please, calm
You
down.
said something. Sorry.
Hello, listen to me. Don't
Sorry.
tell me sorry. Yes? Because you have not done anything.
Okay, sir.
Okay. All right. Then.
Okay.
Yes.
Okay, sir. So in the previous slide, you mentioned something about, food vacuoles being formed from phagocytosis, but I'm actually really confused because
No, no, no,
I
no, no, no.
because I don't understand how they are formed from
Calm down, calm down. Food vacuoles are implants. They are storage.
Okay, sir.
Phagosomes are the process, when a cell takes in solid. Phagocytosis, when the
Yes,
cell
sir.
takes in solid and it has boarded up into, then it becomes a phagosome. This is a food vacuole. And then the
Yes, sir.
lysosome links up with that and digests what is inside and then releases it to the cytoplasm for use. There are
Okay, sir.
several lysosomal pathways. Okay, so have, 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 the laptop is out. Oh God.
Thank you
And
very much, sir.
nobody's going to leave for me to come back in.
Okay.
Okay.
But in the meeting already now. You're in the
Even
meeting already. Right?
No, my phone.
his other
I'm using the
this
phone and I'm using my
His other
laptop for
device.
the
Oh,
laptop. So
okay, I
you try to.
understand.
Well done. Okay, so let's have
But these
questions.
people will live like this. Who will live?
So Toluwani,
Yes, sir.
even if
You
I
cannot
have
still
a
keep
question.
the laptop. People's network is fluctuating, so you might be able to come
Someone
back
has
in.
left. You can enter. Someone has left.
Hello, sir. You
Okay.
can
Sir, you can
practically switch to your system.
I can actually do what?
You can, if you have
You can
the
log in with
Zoom
your
app on your
system.
system, you can switch instead of
Uh,
leaving.
Switch to what?
Yes.
Just
download the
open
Zoom
the
app on your system.
open the Zoom app on your system. Then you see the device you are on. You are in meeting, so you switch. You see the switch button there. You switch.
Yeah.
Okay.
So it's already free.
So if I switch now, then that means I don't have control of my phone, right?
You
No,
still
sir.
have
No. Yes,
control.
sir.
Yes, you
It's already
have control of your
full.
phone if you switch.
Yeah. It will only be your
It's already
laptop,
full.
so you press your screen like that.
All right. We'll see what happens. It's coming. The network is slow.
Sir.
But in the meantime, yes, I'm doing that. In the meantime, ask your question.
Okay, so sir, you said something about the unipods, the sympods, and the antipods. Please,
Yes.
can you?
So ask your question. Can I do what? Hello, Toluwani. Are you the one asking a question? Hello? Where is everybody?
sir.
Somebody was asking a
Maybe-
question just now.
Yeah.
Toluwani,
Traveling
your hand
network.
is up.
Yes, sir. You said something about the unipods, sympods, and antipods. That can you please come again concerning the unipods?
Okay. The transport system is powered by proteins that we call transporters. All right.
Okay. Yes, sir.
They move things across the membrane.
Yes, sir.
They will carry the thing and take it whether from the inside to the outside or from the outside to the inside. If that protein is taking one product, one thing, one molecule from outside to inside, okay, or it's taking it from inside to outside, it is a uniporter. That protein is a uniporter. That type of transport system is a uniport transport system.
Okay, sir.
Means the protein is carrying two things, either from inside to outside or from outside to inside. Okay? And it is doing it either at the same time or one after the other, one immediately after the other. That's why I said sequentially.
Okay, sir.
Okay. Now for antipods, and okay, so for the sympod, The protein that does that is called a symporter. The type of transport system is symport transport system. Now for the antiport, is the protein is carrying two molecules in opposite direction, either at the same time or sequentially, either together or one after the other. Opposite, those two molecules are moving in opposite direction. So that protein is called an antiporter, and the transport system is antiport transport system.
Okay, thank you, sir. Yes, sir. Thank you, sir.
Munachi, no problem. We'll just do questions and answers.
Sir, in the prokaryotic cells, you said that ribosomes has 70S. How did you share it?
How did I share it like how? I'm not
Like
the one that shared it to.
You said that
share it to.
That's the big how would I put it? You said in the eukaryotic cells.
The large subunit.
Yes, sir.
The large subunit and the small subunit.
Yes, sir.
Okay. All right. So prokaryotic cell. The ribosomal subunit, the small ribosomal subunit is 30S. S there is feedback unit. It's a form of density. That means if you have you seen a centrifuge before? If you spin things down, you know in your physics, when you are being taught physics that time in your primary school, you are taught angular velocity, you are taught centripetal force and centrifugal force, and then you are taught escape velocity. If I'm not mistaken. So once the mass corresponds to this centrifugal force, the spinning force, they stay in solution. But if it exceeds, it will go down as a pellet. Because at the end of centrifugation, what you have is the pellet and supernatant at the top. So the feedback unit is a form of a term, a density term. So for the prokaryotic ribosome is made up of two subunits, small subunit, large subunit. Small subunit for prokaryotic ribosome is 30S. The large subunit is 50S. So that means if I have, if this my case was the ribosome made up of two parts, right? Okay, two parts, small and large subunit. So if I broke this thing, if I smash it on the floor, to split it into two. So I will have a small subunit which will sediment at 30S, and I will have a large subunit which is sediment at 50S. But if I have the intact ribosome, it will sediment at 70S. So my math is not so good. I'm sure that's where the confusion is. So the same thing applies to the eukaryotic ribosome. The small subunit will sediment at 40S. The large subunit of eukaryotic will sediment at 60S. So the intact one for prokaryotic will sediment at 80S. So don't think 40 plus 60 should be, should not be 180.
Yes, sir.
Yes.
Thank you, sir.
Any other questions for me?
Joshua.
I believe
some people on YouTube that.
Yes.
Yes, sir.
Question.
Sir, you said that the symport and the antiport can transport two materials into the cell and outside the cell at the same time.
No, I didn't.
But sir, is it possible for?
I did not say that. I did not. That's not what I said.
Okay, sir.
What I said was symport. Okay, yes, symport and antiport. You are saying yes. Symport is transporting both things in one direction. Antiport is transporting both in opposite direction.
Yes, sir. So, sir, my question now is that, is it possible for those transport systems to transport more than two materials in the cell at a time?
More than two? No.
Yes. Okay.
No. Two. Why they call it symport is they are going in the same direction.
Okay.
Why they call it antiport is going in, both of them are going in opposite directions.
Opposite directions.
Yes, that's why it's antipoda. You know, if you go to an hotel, a good hotel, once you come into the hotel and you have registered, you have booked or you have paid, they are going to press a bell, and then somebody comes out with the waistcoat and he carries your bag for you to your room. Those are what people call porters. If that person is carrying 2 luggages for you and heading to your room, that is a steam porter. Again, when you are in the hotel, porter is also supposed to assist you with your baggage. Okay, so Olumide Adebesin.
All right. Good morning, sir.
Morning.
Good morning, sir. All right, sir. I have
morning.
a question. You said something that lysosomes are only present in animal cells. But my question is that what do plant cells use to like carry out all this like autophagy and like cellular digestion?
Yeah, yeah.
So did you get it?
Yes.
Should I go over my question?
Yes, sir.
So can you hear me?
I can hear you. Remember that plant cells, what do? They manufacture things.
Yes, sir.
Animal cells, what do they do really?
They take in already manufactured food, so.
Okay. Right. So you are thinking the lysosome should be an integral part of the plant cell.
Yes, like what would they use to carry out like probably like autophagy or sometimes, if possible, like some cellular digestion as well.
I suppose you have organelles that. I'll write that down. Talk about it.
So,
you remind me of at the next class. Williams.
So you said if based on the previous slide question, you said that. Can you hear me now?
Come up another one.
All right.
Okay.
Okay. Go ahead.
You said that 2 molecules cannot pass with that same pot antipod. But how come there are 2 directions if it's just 1 molecule? Like, it's not.
Can you rephrase your question?
I think some other people should turn on a microphone. That's why you can't hear me.
You know what? Let me just put everybody. Then you now unmute yourself. Yeah. Okay. So you can unmute yourself now.
Okay. So I was asking, since it is antipods and sympod, like they are in like 2 directions, one in the same direction, that is in opposite direction. How come it's just 1 molecule, not 2 molecules?
It's not 1 molecule.
Oh, I thought you said 1 molecule the other time when that
is unipod that transport. That's why it's called unipod transport 1 molecule in whether it's transported outside or it's transported. Sympod will transport 2 molecules in the same direction. That means either from inside to outside or outside to inside. Antipod will move 2 molecules in opposite direction, either simultaneously or sequentially, either at the same time or Are we clear? Giraffe.
Yes, sir. Thank you. Yes, thank you.
Magdalene, please, if you don't, when you want to ask a question, just raise your hand. I will call you.
Okay, good morning, sir.
Yes, morning.
Okay. My question is, David, Ed, can you please highlight the process of the passive transport? Because of your rush, I did not know on the and you said something about symport, antiport, and the other one. Which process is it under? Is it under passive process? Can you please just explain that?
So, passive transport, any form of transportation that does not require and so example is your diffusion. Example is the osmosis. Okay, then we have facilitated diffusion, still under passive. Okay, that one means that something is assisting the movement of those things. Okay, then we have active. Active means that energy is involved. And usually when you talk about active transport, it means that thing is moving things against a concentration gradient. By gradient, I mean there's a place where molecules are concentrated, and as you move further from that area of concentration, it becomes less concentrated. So an analogy will be that if you were in seated on the bus and somebody in front fast, people in front will say, you are the back, will say, why are they saying? Then once you perceive the aroma, you know the reason why it will say. So it means the fat has diffused from the site of origin and has come towards you. That can happen in passive, what's it called? Passive transport. But for facilitated transport, means something must carry. Okay, and if the molecule carrying is utilizing energy, utilizing ATP, then it's active transport. But if it is not utilizing ATP, then it's still passive transport. But a facilitated passive transport. Are you clear?
Yes, sir. Sir, please, I have a question. Sir, can you hear me? Okay,
Yes,
sir, please, I'm asking if this class is being recorded on YouTube.
I'm recording.
Okay. Okay, sir. Thank you.
Are there no people on YouTube?
I don't know. So because your voice was breaking at a point, so I wanted to go back and watch it on YouTube.
And it is not there.
Yes, maybe I didn't get the right name. Sir?
My name is Olumide Adebesin.
Okay, I'll find it, sir.
Olumide Adebesin.
All right, sir. Thank you.
All right. So we'll continue our class on Monday, God willing. And yes. So all right. So we come to the close of the class. Any other questions before we go? All right. So have a lovely weekend.
Thank you, sir.
Someone is waiting.
See you. All right.
Thanks, sir.